pleased that we have Suzanne Dufault, who is here, who's going to give our talk today. You can see the title there. Suzanne, I've lost my bio, but I'll do it from memory. Suzanne is assistant professor here in Department of Biostats. She's worked in clinical trials on a number of different infectious diseases, most recently TB, and her expertise are in sort of clinical trial methodology, different statistical methods. She works across a number of different areas, and so we're very pleased to have her speak here. So thank you very much for that. I think we will take questions at the end. I'll let Suzanne give her talk and really look forward to the discussion. The only other thing I will say is that in front of you, you will see there is a paper highlighting our IGHS theme for the month, which is tuberculosis. You missed grand rounds last week, but there's a couple of other TB focused talks coming up this month, so you're very welcome to come back for those. But I will hand over to Suzanne. Thank you very much, Suzanne.
All right, thanks. Yeah, thanks for having me. I'm really excited to be here. I am presenting work that I started as a TB ramp scholar, hence my UC track slide here, which is supported by the UC track mechanism. Also recommend checking out, if you're unfamiliar with TB, reaching out to the Center for TB portion of the UCSF website, see all the awesome stuff that my colleagues are doing. A lot of them are a part of what I'm showing today in their work, and so hopefully I'll do it justice. Any great innovations, are a team effort, and any mistakes are mine. So happy to take questions and go from there. The topic for today is talking about duration response, randomized design. So a quick show of hands, how many people have heard about something like this? Okay, all right, we've got some hands in the air. Great. Well, so I'm looking forward to, maybe doing some brainstorming at the end, some discussion, and hopefully for those who didn't raise their hands, you'll feel more comfortable with what that concept is and maybe where the field may be going. And ultimately, I'm going to frame this in the case study of finding a shorter duration of TB treatment. If you're not familiar with TB, I'll give you a little background, although there are experts far more familiar with this topic in the room. But I'll give a brief background on why we need a shorter duration of TB treatment. But the concepts we're talking about can be mapped onto a wide variety of areas, not least of which antibiotic duration use for general antibiotic use, but I think a number of other areas as well. So I'm interested to hear kind of how you think this pairs with your own work. Okay, so brief roadmap, like I said, I'll do a really brief state of TB treatment just to kind of get us motivated. And then I'll talk through an example of a traditional duration response trial design, just so you know kind of how things have been done in the last decade or so. And then I'll propose this alternative design for duration response trials. We'll ask questions, is it more efficient? Is it more accurate? Can we do it earlier in the trial pipeline? And then finally, give a brief, not fully fleshed out list, but some upcoming and ongoing duration response trials that you might want to keep your eye on to see if we can identify shorter durations of TB treatment. And again, this is being used in many fields, not just TB, but I'm going to highlight TB for the sake of this talk. Okay, so TB quick facts, right? So if you're unfamiliar, maybe today's the day that you walk away knowing that this is, one of the most deadly infectious diseases that we have circulating, and that's been true for centuries on millennia. Right, and so 1 in 4 of the global population is likely infected with TB. I can't quite see there. Okay, and more than 10 million, are estimated to fall ill with TB annually. Okay, and you can see here is the map just marking the incidence. You can see it's not globally spread in an even manner, right? There are kind of hotspots. There are a couple of countries that have a lot of incidents. Some other countries have a little less, right? And so this is a geographical problem, but it is something that we're seeing right now in our backyard, right? We're seeing these cases in SF and in Kansas, and we've got great experts in the room for talking about what the impact of that kind of transmission is, right? But it's really widespread. It's something we should all be aware of. Fortunately, it is treatable, it is curable, and for those who have drugs susceptible to TB, right, we have an 88% cure rate. So it kind of boggles the mind, right, that this continues to be the #1 infectious disease. Above HIV, above COVID, right, above your favorite priority disease, right? Well, I think there are obviously a lot of complex forces that go into that. and I'm not going to touch on them now, again, because we're focusing on treatment. But what I'm going to talk through next is kind of why the treatment of TB. So just because you fall ill with drug-susceptible TB and we have regimens that work, there are a lot of reasons why you may find it hard to complete treatment. So let me show my next slide here. Okay, this is a figure that I took from an activist guide to shorter treatment for drug-resistant TB. But basically, I like it because it highlights the treatment landscape. And I recognize this text is really small. But basically, there's drug-susceptible TB. And if you have that, we have kind of three main options, right? There's also, you know, preventive measures for if you've got an infection and we don't want you to get disease. And then there's varying degrees of drug-resistant TB, which require longer courses of treatment. And I'm not going to go into all of them. I'm just going to focus on the drug-susceptible TB right now. And so as you can see, there are kind of three main regimens that are in use, or at least approved for use. There's the standard of care regimen is the 6-month standard regimen, which is comprised of 4 different drugs. And I've learned, because I'm new-ish to this, that doesn't mean that's 4 pills, right? That could be upwards of 20 pills, I think, you know, per day that you're having to take for a duration of 6 months, you know, and that changes over the course. So there's kind of an intensive phase at the beginning of your treatment, and then it decreases over time. But even still, just like visualizing what that pill burden might look like, which this is not even comprehensive enough, over a long period of time means that a lot of people may not finish treatment. They may not be adherent to treatment, right? And this isn't just on the patients, right? These drugs are also really maybe hard to not only like procure, but then you have them, there's a lot of toxicities associated, right? The longer you're on antibiotics, the higher your risk for kind of patient harms. And so there's a lot of reasons why we might want to say, you know what, 6 months of antibiotics for these patients is probably not the ideal course of treatment for TB. And so historically, right, there have been a lot of ways that we've kind of tried to come to this optimal duration of treatment. And so I think a lot of the initial work kind of focused on these settings where there was high TB incidence, a high TB burden, and they would compare notes, you know, if I treat patients for 10 months, you know, how are their outcomes? If I treat for 6, it wasn't necessarily done in a clinical trial style. It was a little more pragmatic, a little more existing records-based, right? But in recent decades, we've started to try to put this into the state of a clinical trial. And so fortunately, we do have these 2 4-month regimens as well for drug-susceptible disease. I'll focus on the Study 31 regimen, and that came out of a clinical trial where we were comparing, right? But the team was comparing a 4-month regimen against the 6-month standard of care, found it to be not inferior, found it to be not inferior, correct. Okay, we'll talk about this more, you know, but anyway, that's one new regimen over maybe decades, right? This is probably not our most efficient way to find the optimal duration. And so progress has been made. A lot of effort has gone into this. And thanks to those efforts, we can build on that backbone and do maybe more efficient things in the future. Part of that is moving into more efficient duration of therapy studies. They're relatively novel. When I started this work, I did kind of a lit search, and I was looking, right, for anywhere where we were doing, you know, Z-packs. How did we go from 14 to 7 days, right? How do we think about treating people with antibiotics and the duration they should use? And these kind of efficient designs are really not implemented, at least not that I could find. I'd love to hear if you know alternatives. But we've got kind of a future ahead of us where innovation is possible, right? And one of the things that I've already stumbled over is this non-inferiority testing, right? So not only is it a little bit tricky to communicate, it also kind of increases a lot of maybe challenges in the way we perform estimation and inference, the way we make decisions. And of course, it comes with the risks such as, right, this biocreep, right, or the idea that if we're constantly finding regimens that are a little bit inferior, but not outside of a reliable margin, as we're shown here, after K trials, we may end up with something that just really doesn't have the same benefit as our original, right? So it's kind of 2 separate concepts, but I just want to flag them immediately so that we're thinking about them as we think about the next stage of clinical trial. Okay. I mentioned on that previous slide that we do have a 4-month regimen, right? And that came out of the study 31 work. So this was a large phase 3 study, and it found a 4-month regimen not inferior to the standard of care 6-month regimen. And I want to use this as an example of how maybe traditionally, but that's not quite right? But how recently we've been trying to answer the question of the optimal duration. And so here I've got the study schematic, which is from their published protocol. And the basic idea, right, is this looks like any other phase 3 parallel arm trial, right? I've got my enrollment criteria, and then I randomize 1-to-1, and I'm randomizing, in this case, between 3 regimens, the 26-week standard of care, right, and then 2 17-week regimens that don't have the same drugs, right, but they are shorter. And the hope was that we could identify one of these to be not inferior to the control. Okay. I think this is great. One of the challenges, right, is that, well, I'll get into it. You know what? I'll let the next slide speak for itself. But that's just a little background to how that study was done, how we got to our existing 4-month regimen for drug susceptible TB. I think the question I want to pose, right, can we do earlier, more efficient duration ranging studies? This was a large phase 3. There were 3 arms. There were hundreds of participants enrolled. It took many years, right? And at the end of the day, we had one of these 2 promising regimens actually meet the criteria set to be considered not inferior. The other one was close. Could we have done that more efficiently? Could we do it better? Right. So for targeting this question, I want to start with the more efficient duration ranging study first. As I said, right, a parallel arm study, a traditional perhaps framework for doing this would be to, you know, randomize to 2+ arms. Maybe they have the same drugs, maybe they don't, right? And then put that within enough. So randomize, right, 1-to-1 to these different durations on these different drugs, and then do a non-inferiority comparison. Say, okay, let's compare regimen 2 to regimen 1. Is that not inferior? Let's compare regimen 3 to regimen 1. Is that not inferior? I don't know. You know, we will find out. But I think that immediately raises 2 key questions. And these are really well illustrated in one of the references I'll cite later. But question 1, if this is a phase 3 trial, right, a lot of other work has been done. It's probably gone through phase 2 studies. How are we making that decision for 17 weeks of treatment, right? Why 17 weeks? Why not 18? Why not 14? What are we basing that off of? And this is clearly a critical decision because we're never going to do this study again with a different duration of treatment, right? So that's a really important question that I don't know that we have. Clearly, we don't quite have the way to map that really efficiently, right? And then the second one is, should we treat these different durations of maybe the same treatment as independent? So in this study, we were looking at 2 separate arms. I say we, this was not me, you know, but the field was looking at 2 separate arms. They did not have the same drugs. But what if we did have multiple durations of the same multi-drug regimen? Would we treat each of those as independent observations, or could we learn something, right, from a shared assumption about how those duration response curves, as a bit of a signal, right, should operate? I don't think that's necessarily clear if we're applying kind of a traditional parallel arm study where we do make this assumption that these arms are independent. So here I'll walk through the proposal. So this goes by many names, and I will unfortunately use them interchangeably, but I'm calling it, on the one hand, this is a duration response design. This is a design framework that I think works really well for evaluating duration. It's also referred to as the durations design. That's how it started, but now it's been rebranded as the response over continuous intervention design. Again, a little bit unfortunate, but it makes it clear that anytime you have this exposure that has some kind of continuous element, right, if we can randomize over the continuous range, we might be able to learn something interesting instead of treating them as independent or separate units of observation. So unlike the or maybe very similar to the standard design, right, we are still doing randomization, But now instead of to 2+ arms where we pre-selected duration, we're maybe randomizing to a lot of durations from the same multi-drug regimen, right, within a reasonable range. And this is really tricky because we want to learn as much about the duration response as possible. And if you think that through, that means we have to randomize some people to shorter durations that may not be as effective. And we also want to randomize people to longer durations that are certainly effective so we can learn that curve. So this sounds simple, but in practice, I think it's the point of a lot of debate and contention. So we can get into that in the discussion perhaps. But basically, in theory, it should work really well. We're going to randomize across a reasonable range of treatment durations. And then I'm going to fit a model, right, across those multiple durations so that I'm learning about the drugs or multi-drug combinations duration response, just the same way we would with dose response, for example, right? Okay. And as a simple note, if you're like, but I love non-inferiority, and that's what we should do, you can still do it. Like here, fitting a model does not prevent you from using any of your favorite hypothesis testing frameworks. So flag that for later. We'll talk about that too. Okay, so side by side, if I've got my kind of traditional, randomized to 2+ arms, do a non-inferiority pairwise comparison trial versus my ROCE or duration response design, right? Now these two questions that I flagged earlier about how are we picking those optimal durations for comparison and should we treat different durations the same treatment as intermittent, I say have really easy answers, right? On the one sense, it's easier to say, well, I think that this range, right? And even the modeling and whatnot that we do to get to that hypothesized optimal duration, right, is probably more likely to come with a range of uncertainty. So it's a little easier to say, how do we pick the range of durations for comparison? That's a little more straightforward. And then how do we treat the different durations? Well, we're going to treat them as related, right? They're observations across this continuous exposure. We can assume that there's some relationship there. And those assumptions, depending on the method, can be really formalized. And that can be a good part of discussion as well. So I won't necessarily get into that, but I'm happy to talk modeling strategies as well. Okay. So then the question is, why should we do something totally different if it gives us the same results, right? This obviously is a little more complicated. We're randomizing among many arms, and we're having to do all this observation and model specification. Why should we change? I would say, well, let's ask what it can bring us. Is it more efficient than traditional methods? And here I'm sort of citing some of the work that our colleague colleague at UCL, led, along with kind of Matteo Cortagno, who kind of initialized, the durations design. And they designed a simulation study, in this case, to identify the best design to assess whether any regimen can achieve a similar efficacy to the standard of care without being as long. That's a lot of words, right? But just like, what is the best way to design a study so that we can answer this question of is there a shorter regimen, right, than the one we currently have? Okay, and in the simulation study, they looked at a range. We won't dwell on this, right? But they looked across a range of parameters. So specifying different event risks, different durations of novel treatment regimens. Should we be looking at 5 different durations across that duration response curve? Should we be looking at 3? How many people do we need assigned to each of those durations? And what are the shapes, et cetera, different non-inferiority margins. I recommend, if this is interesting to you, check out this publication because it's pretty well written. I agree, very insightful. Okay, and I'm just going to pull out kind of one of the key results that I think asks or answers this question of, is it more efficient? So this is one of their key results tables in the leftmost column, right? We have an observation of 40 participants per arm. Again, that's the randomized duration, right? So total sample size of 500, we've observed 5 different durations of this multi-drug regimen, 40 per duration. We've got different event risks, among the standard of care and non-inferiority margin. And what I really want to call out here for efficiency, when we're thinking about efficiency, right, we're thinking about decreasing the variability of our estimates, which leads to increased power to detect a difference if it's there, right? And I'll draw you first to this first row. The no model is basically saying, let's do things the way we've done it, right? Let's do those pairwise non-inferiority comparisons with each duration against the standard of care. The power in this setting, 80%. Okay, that's great. But we can see that if we start to treat this as a response over continuous intervention trial, and we start to learn across those durations, our power to detect that one of those durations of our novel regimen is not non-inferior to control, it goes up, right? We have increased power, suggesting we could, decrease sample size, or if there's a lot of decisions we can make based on that. And the authors, they conclude, right? They ask themselves directly, is modeling the duration response curve necessary for improving a trial's operating characteristics in the setting? And their takeaway was power is always higher, which I don't think is super surprising to any of us, but it's always higher when we're using a model to estimate this duration response relationship. So, and in this setting, they didn't do any kind of interpolation necessarily, right? They used the model to fit this curve, and then they still did kind of a testing procedure where they were only going to end up recommending a shorter duration if it was one observed. So do you see what I'm saying? So even if the optimal duration was, say, 9 weeks of treatment, they would say the 10-week observed duration of treatment, if they observed 10 and 8 weeks, they'd say the 10-week was the duration they recommend, right? So we're using that curve to kind of improve our precision, but we're not using the curve to, say, recommend a duration that wasn't observed. Right? There are ways we can do this well. But that made me think, well, okay, but what if I want to know the actual optimal duration, right? Like, what if I don't want to recommend just the one that we observed? I believe that this duration response curve is real. I want to be able to recommend durations that weren't maybe directly observed, but would have been the best one for this multi-drug regimen, right? And so is this response over continuous intervention more accurate, for example? And this work is published in our paper in Trials. So leave it in here so you can look it up and play. But we had kind of two goals, which one was to adopt a lot of the methodology from dose ranging, right, where they've kind of figured this out a little bit, right, and see if we could use it for duration ranging. And then also see, could we be more accurate with our estimation if we adopt some of these approaches, instead of kind of relying on the traditional ones. And so, again, we do a simulation study. And if you haven't seen simulation studies before, or maybe you have, but I love simulation studies. So if you want to talk, I want to talk. But I'm like, the beauty of a simulation study, especially when you're doing methods development, right, is you know the truth. You know exactly what the truth is. And so we simulated 3 different curves that we thought could be plausible, and this is a great place to have a lot of discussion with collaborators, right? If I'm looking at different durations of treatment, is it reasonable to assume that 8 weeks would give me 85% efficacy, right? You can have these kind of tailored discussions if you would like, if you're designing a specific study. Anyway, we set up a curve, for example, like this, where we've got a logistic growth relationship, where at the lowest end of duration, we've got 85%, probability of cure, at the top 95% probability of cure. And we can estimate, we know the truth, right? We know that there's an effect. And so in our simulation study evaluation of our methods, we can assess whether they detect evidence of an effect. We know that by truth, it should be there, right? We should also be able to see how accurate is our modeling at identifying the actual correct duration response shape, right? Which might give credibility to any interpolation we want to do. If we can properly identify the correct relationship, we should feel a little more comfortable about interpolating between observed doses. And we can specify what is the actual minimum effective duration. So in this case, I just said, let's let this be the actual duration that corresponds to 90% probability of cure. Wouldn't have to be that. But this was the setup. And I'm going to show you some results. Okay, I see they're not super clear for the room, but I'm going to draw your eye first here. Thankfully, you don't have to be able to see the axes that well to get to the story. I think it's pretty clear. Is this design more accurate at estimating that 10.2 actual duration of minimum effective duration? On the bottom, these 2 lines here are doing a non-inferiority pairwise testing, right? And we almost never get something within a reasonable range, right? And part of that is because non-inferiority testing is making us a little more conservative than I think we realize, right? So even when we have a regimen that passes, it's still probably being given for too long, right? So that's here at the bottom. This is the probability of successfully estimating the minimum effective duration within an acceptable range around the truth. So complicated metric, not that complicated. And then up here are all of these different modeling procedures that I borrowed from dose ranging, right? Dose finding studies. So we've got fractional polynomials, we've got linear splines, we've got NCP mod, which is a personal favorite, and so forth. But at 100 participants, the gap is incredibly wide, right? We can be more accurate. Again, that makes sense. It feels like common sense there. Okay, they're far more accurate at estimating the minimum effective duration. Can I get a time shout out? I can see the clocks.
It's 12:32.
Okay, so I'm doing okay. Yeah. All right, awesome. Okay, good. So that was kind of the, can we do it more efficiently, right? We can be more accurate and we can do a little better with our ability to estimate precision, right? Result increasing our power, right? So then I feel like the natural next question is like, can we do this earlier, right? Do we have to wait until phase 3, mobilize all these resources, mobilize all this effort and all the years, or can we start doing this work earlier in that clinical trial pipeline? And so I'm going to move this work into the phase 2 setting, right? So we're thinking about, does it work? And now I want to throw in there, at what durations, right? That's the second question. Okay, so I'm bringing you back to some of the results from that earlier paper that I mentioned that's in trials. And again, I apologize, it's a little hard to see in the room, but I want to bring your eye to this side of the axis now, right? Where again, the gap is evident, right? We can do pretty decent estimation of the true minimum effective duration, even at sample sizes. What I've got on the x-axis here are sample sizes per duration. So when we're randomizing, say, 30 per duration, we're already at 60% probability of estimating within this window. Maybe that's not high enough. That's a debate to be had, right, by the design team, especially since now we're looking at it in a phase 2 context, right? And so you're kind of trying to get evidence suggestive of what to put forward. But I think it's a lot further than we would have been if we just tried to stick with the traditional approach, and could potentially be resource saving. So, and if you're like, okay, great, but what do the curves look like? Again, in this room, I don't know, with this light, I think it's getting really, it's okay? Okay, maybe I'm too close. So here's what the curves kind of look like. And so I'm not saying that we can do this with no participants, I think is my point. So on this top row here, this is 10 per duration, right? If I'm fitting that duration response curve, the black line is the truth, right? And each individual line is an estimate from a simulated data set where there's 10 per duration. At 10, it's a little chaotic. I don't think I would ever recommend going quite this low, right? But you can see how that improves when we just increase sample size to 30, right? And so it's nice to be able to visually see that when we're doing 30 per sample size, we're pretty accurately covering the truth. It's not exact. That's what our confidence intervals and so on are for, right? For helping us a little bit, right? And there are ways that we can increase maybe the conservative metric we're using by which to propose a method forward. But still, I think we could do this more efficiently, more accurately, and earlier, right? To be in a better position to move into phase 3. Okay. So I'm just going to point out that we do have new work in writing right now in the manuscript phase, where we're looking at other complications related. And so if you attended my talk in the Division of Biostats before the holidays, I talked about this a little bit, but where we're thinking about, what are these maybe improved ways that we could handle small sample size challenges, right? If we're going to be moving into a small sample size space, can we do that more efficiently by using things like effort correction to handle complete separation in that setting, and so forth? Happy to talk about this. And then also, we do have to be thinking about intercurrent events, right? Anytime we're talking about TB. So if you're unfamiliar, right, intercurrent events are things that happen between randomization and observation of your endpoint, right, that are likely informative, right, in some way and otherwise uncaptured. And so in TB studies where these are really long and we're watching people over a long period of time, not having a plan for handling intercurrent events is a problem, right? And so we've got some work coming out that shows that once we handle these things too, we can get even better results. Okay, and then I think this is kind of my last set, is looking at just a couple to put on your radar if you're watching this space. So some ongoing and upcoming response over continuous intervention trials, or trials using this design. The first one I'll flag is the paradigm for TB study, and we've got the NCT number here. But this is one that luckily, Patrick, myself, and many others get to be involved in. We are working with the team at UCL, and that paper that I mentioned earlier, led by Tra, that was really the design paper that kind of explains the rationale for this phase 2C trial. And I guess I should mention first that this is kind of cool because it's a multi-arm, multi-stage trial, right? So we first did this regimen selection phase 2B, and then those winners, right, are going to get sent on to duration ranging phase 2C, hopefully leading to some really exciting phase 3s very soon. The dramatic trial, again, a 4-arm phase 2, and again, note, these are all happening earlier in that pipeline, like we said, right? And so here again, they're looking at MDR TB participants and looking at, could we do treatment much shorter than the current standard, which is well over 6 months, but also quite a bit longer, depending on the extensiveness of your MDR TB, multi-drug resistant TB, right? And then last, Spectra TB, which is of course led by Gustavo in this group. And so really great work that's in preparation, looking at treatment shortening and stratified medicine, right? So it's a fun way to separate out high and low risk and maybe do some treatment shortening among those who are easier to treat for drug susceptible TB. So my last slide, maybe I'll leave you with this. This stood out to me when I was trying to figure out, like how dose ranging has evolved, what we can learn from them, what we can do different in duration ranging. And they had a workshop in 2014 where the kind of right up after the workshop, it read awesome. It's one of those where you're reading it and you're like, I feel energized, And one of the key quotes that stood out was, selection of dose for phase 3 is an estimation problem, should not be addressed via hypothesis testing. And I think we could say that about duration as well, and maybe many other continuous aspects of our trials made me excited. Thank you. I'll pause and open for.
Great, thank you, Suzanne. So, questions for Suzanne?
I have one online, but I'm seeking clarity. I think that they were referring to paradigm. What drug doses or treatment regimens were used in this study, and how do they compare with the standard WHO recommended BPOM or BPAL regimens, doses, and duration administered over 6 months? Do you want to read it?
Yeah, it's not clear what trial they're referring to.
Yeah, I just asked them. They're anonymous.
Okay, well, for a little clarification, maybe.
Yeah.
So paradigm is targeting drug susceptible TB, right? And so that is true. All the regimens are being compared to the 6-month standard of care, HRZE, right? There's also in that trial, I think, the evaluation of a 3-drug combination, BPM, I believe. But that's about right. So in terms of which ones will be evaluated and which ones will win, that I can't necessarily say, right? It's ongoing right now. And so the exciting thing, which this is published on clinicaltrials.gov, but they're looking at these multi-drug combinations where maybe the backbone, the 3-drug backbone is standard, right? It's drugs that have already been approved, including bedaquiline, protominate, et cetera. And then the 4th drug is a novel drug, right? And so we've got 2 in this first wave. Again, this is all on clinicaltrials.gov, but we've got a BTZ-043, if you've heard of that. I don't know if that has a name yet. Okay, BTZ-043, and then also a compound from GSK, which does have a name,
Ganfepperol.
Yeah, Ganfepperol. And so it's a lot of different multi-drug combinations of those kind of 2 novel drugs and a 3-drug backbone. Yeah, it's exciting. We just had the IDMC for the regimen selection portion, but there's a lot of work left to do, so I can't spoil anything.
Yeah, thanks, Suzanne. I think that was the answer to the question. Yeah, Priya, go ahead.
Thanks, Suzanne. It's really interesting, and I'm certain I did not really understand a lot of it, but that's good. I'm learning. My question is, how, and we can phone a friend or whatever, like, how do these methods and simulations, how are they reviewed by regulators and policymakers when they're making decisions for guidelines and for national strategic plans and operational work plans? Like, can you describe a little bit maybe from TB or from other diseases, that process?
Yeah. So I think, I'll take a first stab, and then you can tell me if I'm on the right track for your question. Yeah. So for simulations, I really use them in the design phase, right? And so it's kind of the procedure that we, or at least I've used historically, to set up the trial to kind of maximize the opportunity for success if it is there, right? That's critical. If it is there, we want it to be successful. And so in that case, it's gone over really well, right? And so for the paradigm trial, for example, for the regimen selection, we did a really large simulation study where we looked at, first, we learned about maybe relationships. And again, this is the regimen selection. So this is not the duration element. We're just looking at how are we going to select the most promising regimen. And in that case, I looked at relationships between our key primary biomarker endpoints that decisions would be made based off of. I looked at relationships of those biomarkers in existing trial data. So specifically the Remox data, I tried to learn from that. And then I built a simulation study to say, if we saw something similar, or if we saw something worse or better than how those regimens performed, right? How well would my method do at selecting, or how many people do I need per arm to have the best chance? And so in that sense, I think simulations, maybe they happen much earlier than the conversations with regulatory bodies, but they are useful for setting up, how to design the trial.
Yeah, so it sounds like there can be a really efficient, formative piece of work, and that it sounds like regulators who go back and see that the design was informed by simulations are just as likely to kind of view the evidence generated from those later trials as they would if there were some pilot or something else.
I think so. I think even if you had pilot data, I don't think that would necessarily mean you wouldn't do simulation, right? That would almost be ideal, because then you could feed that directly in to your simulation, right? And project it out, if we saw the same thing we saw in our pilot or more or less promising, right? And so, and I think one of the things, again, on the last kind of big phase 3 that I was involved in, which was the Applying Wolbachia Eliminate Dengue study, where we're looking at like the intervention being a mosquito-based intervention, and we're looking at the prevention of dengue incidents in various clusters. Anyway, without getting into details, we had a lot of rich historic data of dengue incidents in this area. And so we were able to first design the study based on what if our intervention has this impact? In all these historic periods, what might we have observed, right? Would we have seen that impact, persisted throughout the data itself or not, right? So we were able to set up kind of like historically, if our intervention works at this rate, what will we have observed? Project out. And then at the end of the study, when we saw that it worked at such a high level, we were able to point regulators back and say, yeah, we looked at kind of every possible scenario, and this kind of, jives with what we were thinking might happen, and so in that sense, it just kind of added on. It's very helpful.
Let me ask. Oh, yeah, go ahead, Sophie. Yeah, please do.
Well, I was going to say, I'm curious. So this is kind of sort of feeding into how you design the studies, but I know you and others here at UCSF have also worked on when you're actually doing the study. How can you be more efficient in disregarding arms that are underperforming, and how does that how does this method sort of interplay with those methods?
Yeah, I think there's more work to be done there, to be frank, and so one of the nice things, again, your simulation study can be as simple or as robust as possible. And so for the paradigm study, I was designing the regimen selection part, right? We were evaluating our ability to detect the promising regimens during an interim analysis using this biomarker, right? And we simulated all the ways that might be done. And that gave credibility to the way we were going to make decisions or propose that, the trial steering committee makes decisions. Then we could have simulated, okay, well then what happens next? What happens after that? And that would be kind of a two-stage design, which I think would be kind of more informative as to, the impact for secondary post-decision, right, performance, but we did not do that, right? Yeah.
The next
step, yeah, the next wave,
next few years of your career.
Yeah, Ben.
Suzanne, Ben Arnold at Proctor Foundation. That was really nice. I'll just mention Suzanne gave us some really great advice in designing a study using these methods. We haven't started it yet, but in a totally different context. And as we've thought about that design, I was curious, I have a follow-on question actually. Is there any guidance from the TB community in this context on interim analysis? Because for example, an optimal duration, if you're using interpolation, that's not a test statistic. Often you're trying to get some parameter and maybe uncertainty around it. And that's okay. We can design studies around precision of something we care about. But it's oftentimes with our interim analyses where we have pre-specified guidance for stopping early, for efficacy or futility, we often base that on a test statistic. And so now, we're sort of faced with what will we think about on the interim? Is there any guidance yet around interim analyses in this case? It sounds like there might be.
Well, around interim analyses, I'm not sure on the duration side. We do have, obviously, right, in the statistical analysis plan of the protocol for Paradigm, they have specified how they will look at the duration ranging during an interim, but I'm blanking on how they're proposing to do that.
No, I think it's not formal rules, it's more looking at the durations. And if there are too short durations that are looking unsafe, then they're stopped. Or, if the durations that are too short have too low efficacy effectively, then they're stopped, or it's more that guidance. But I mean, maybe so I think one of the things you've done, Suzanne, has been interesting is really getting familiar with the dose ranging literature. And I don't know if there's any learnings from the sort of dose finding literature in that area. I don't know if you've got any reflections on that. Okay. Well, I guess one specific area is use the control, how that is one difference. I guess that's in dose finding, you don't often have a control. You're looking at a zero dose, whereas obviously here you often have an active control. And I know you've done work in that area. What are the differences when you have a control in that? And maybe then relates to how does that impact on your interim analyses as well?
Yeah, that's an excellent point. I think, right, so in dose finding, right, it's often placebo controlled, but not always, right? And sometimes there is an active control and the methods exist, and I think they could be interesting and incorporated into the space as well, but it comes with all sorts of challenges about knowing kind of how to do that comparison. And so we did recently, in that work that I'm talking about that's being written up, look at how do we put, the HRZE regimen somewhere on the duration response curve? That doesn't feel quite right because the duration response curve is for a novel regimen that's not HRZE. How do you learn the position of that control? Does it belong at 26 weeks truly if we're trying to anchor, an active novel regimen? And so at this point, I think it's still an open question. Right now, we've got kind of this initial comparison step of doing kind of a simple non-inferiority test, and really kind of trying to use it. I think I would prefer using it in this kind of more holistic way, where if I can reflect back on maybe how we're doing the interim decision making for the paradigm regimen selection, we were really adamant not to base it on a single metric and instead propose this Bayesian multi-metric framework where we're looking at all sorts of things, right? We're looking at how steep is the slope relative to control, but also posterior probabilities that it is actually steeper than control, right? And posterior probabilities that it ranks in the top of its asset class, and kind of trying to pull this almost dashboard together for a more holistic comparison. And that has gone over pretty well so far, especially because we did multiple conversations with, right, the people who need to make that decision and trying to really incorporate their feedback of what kind of questions do they need answered to feel confident. And so I think, you know, having that data on a control, even if we're not yet sure of like the proper way to incorporate it into a single estimate or framework for estimation, I think having it alongside for holistic review would be helpful. Yeah.
Yeah, so there's a question to Sarah, sorry, and then second there. So go first, Sarah.
Really great for someone outside the field, but my question is about the assumptions and the duration response. One thing, are you treating all the patients as if they're all responding the same way, or is there room to start to understand the heterogeneity of slow and fast responders, and you know, at which point you start to decide there are different subpopulations and beginning sort of different directions?
Yes, awesome. Yeah. So this is one of the things that is on my kind of next up / already thinking a little bit. So fortunately, because you're fitting a model, right? Because you're fitting a model to this curve, and either you're fitting a library of models, like in the MCP mod, you're saying, I don't really know what this duration response shape looks like, but I can hypothesize it maybe looks like one of these things, and I'm going to do some kind of aggregate way. Anyway, try not to get in the weeds, but you know, because you are doing model fitting, you can adjust for confounders, other things that maybe are affecting, right, a person's duration response relationship. I do think it gets complicated quick, right, in terms of interpretation, in terms of then how does that convert into a policy, right, where it's a little easier to have a treatment policy if I say, these people are quick responders, they need short durations, and these people are, you know, instead of having this kind of adjustment set. So I think that's an open question. It's one of the things we're going to get some data from the Spectra trial on, right, because they are doing that risk stratification first, and then we'll have kind of the duration response, kind of a curve in the easy to treat, and we'll have duration, 2 points of duration, I think, in the hard to treat people, you know, so it's not enough to make a curve, but we'll have some information there. But if anyone wants to talk or brainstorm more on this, this is like in my next to do category.
Thanks for the brilliant presentation. I just have a quick question about model. So you did a lot of simulation. So I just wonder what kind of model you used for the simulation. Did you use classic statistical model, or did you use pharmacometric PD model?
That's a good question. So in this case, I use classic parametric modeling, right? So it's like either specifying, oh, I think this is a logistic curve, you know, here's my parameterization of that. But I think that's an interesting route that a person could go. It's just not my background. So I tend to lie more in the parametric modeling space. Yeah.
So just wonder in the model, if it's a statistical model, what kind of the other variables you included other than duration?
Good. Yeah. So in the work that I showed today, I only looked at duration, but obviously that's way too simple, right? And so in the work that we're writing up now, we included a couple of things. I might even have the DAG handy if we have. I mean, this is a messy slide, but here's the DAG you can see here. So here I considered X is duration, right? Y is response. And then I considered covariates like age and sex, location, meaning like different sites, right? When we're doing these multi-site trials, different sites have different response rates to the treatment. So location matters. And then your last observed culture result, right? That also impacts whether or not you're probably going to respond positive. And then I also considered, like I said, these intercurrent events, things that would prevent me from observing your endpoint, including, say, death, you know, reinfection from an exogenous source, and then treatment extension. So this is a limited set, but we hoped that it kind of covered several of the main issues that might be driving differences. And at minimum, it's hopefully a chance for us to see how susceptible we are to getting this wrong, right? If we're doing a model and we're getting these things wrong, it's all lost, you know? Yeah.
Any final questions? Yeah, go ahead. Oh, no, you go ahead here, and then Zira has the chance. But go ahead, please do.
Okay. Thank you for a really brilliant presentation and for your help with Ben and us at Proctor. Kind of going back to the population heterogeneity concept, we were hoping to like stratify our randomization by certain characteristics, but I think I'm still conceptually not super clear on how that would kind of play into some of those implications and how that would impact future analyses based on like the curve that you get.
No, I think that's fair, right? I don't know that I have an answer, but I would love to brainstorm more. I mean, one of the things that I want to experiment with, right, is this idea that of course we can learn across durations, right, about the duration response. I think to keep this in a phase 2 space, we also have to think about how we can learn across strata. And that part I don't yet have sorted out, right, in a way that feels appropriate to the design, like you're doing a stratified design. So I'm just flagging that. That's something I'm thinking about. And if you've thought about this or seen it, I'd love to hear. Yeah.
But the data you get from phase 2 can inform your decision for a phase 3 design that brings that stratification, because you'll gather that information and you'll have 2 shorter durations for the low burden of disease and 2 longer durations for the longer, higher burden.
I think that'd be amazing. Yeah.
You get that from this design.
Yeah, you would. You're right. Yeah.
Yeah, it's well suited to phase 2, I agree. Maybe Ziran, the last question. Thank you.
So we have the treatment duration, the best treatment duration depends on the regimen. For new regimen, how many treatment durations do you need and each treatment? And so that ongoing trials, they choose different multiple treatment durations. So why did you choose 10? So we know for participants in the short treatment duration, they are more likely to have lower Suzanne's treatment rate. So how to balance it? We don't want to put patients into high risk.
I think that's a good question. I think if you're designing something like this, you will have to do a simulation study, right, to reflect the expected relationships. So it's hard to make a general comment, but the work by led by Tra, and in the contemporary clinical trials, they do make some general recommendations, right? They found for the studies they looked at that 5 different durations across the duration response curve was kind of better than 3 when you have a non-flat curve. But, and I think we're finding that for the scenarios that we're looking at, that, 30 seems to be a lower bound for what you'd like there. It is better, But all of this is complicated by the point you just made that people don't want to randomize people to, and also we have an ethical obligation to treat people, right? Especially when a treatment exists. So it's complicated. It's really hard to convince people that for science, we need to observe the curve, right? If we only are looking at 5 durations, and they're all in the flat part, successful plateau of the curve, that cannot help us, right? Learn about lower. I don't know how to solve that problem, right? I would not try to come off callous, but it doesn't matter how many durations or how many people you look at. If it's on the plateau, we cannot learn about the optimal duration. Hopefully you would learn that the shortest duration on that curve is sufficient, but yeah, it depends on kind of willingness to explore potentially lower.
Thanks, Suzanne. Thanks, Ziran. I think we'll finish there. That's great. Thank you very much. It's great.